Um, Greetings, ladies and gentlemen. Today we'll be discussing the corn snail's venom, which are conotoxins, various conotoxins that are usually used by the tropical corn snails to catch their prey. Firstly, we'll look at what a tropical corn snail is, in which the tropical corn snail is scientifically called Conus geographus. Conus is the genus and geographus is the species. It is commonly familiar by the name of geographic cones. These species are diverse carnivorous predators, which are divided into three groups: worm hunters, vermivorous, mollusk hunters, molluscivorous, and fish hunters, piscivorous. As you can see, they are named in a way they are divided into three groups because of the way in which they do what they eat, in the way they behave in terms of their feeding habits. Like, for example, fish hunters, they eat fish, so they are called what? Piscivorous. Okay, then genus Conus is their greatest single genus of venomous species with about 500 species. So this genus Conus, it has about 500 species, so it's a very much diverse genus. Looking at the species Geographicus, Conus geographus, it uses a variety of conotoxins, with also activities that vary in different vertebrate classes and their actions in different synapses of the same animal. So you'll find that in the same animal, there will be a variation in terms of activities that they'll be doing in the synapses. These species are found in tropical regions. Then the venomous carnivorous predators vary according to their feeding behaviors, which has already been explained. Looking here, this is a tropical corn snail. That opening is whereby the prey enters after it has been paralyzed by the conotoxin. Then looking at the mechanism of action, how it works, how the whole venom or the conotoxin work in the nervous system. The conotoxins, they do what they target by hindering the closing. They target their prey by hindering the closing mechanism of the ligand-gated, voltage-gated calcium channels, potassium channels, and sodium voltage-gated channels. These predators, they produce various kinds of toxic pesticides or conotoxins, which assault the muscular systems of their prey. The presynaptic conotoxins impaired the voltage-activated entrance of calcium into the nerve terminal and release of acetylcholine. So here we see that the release of acetylcholine, which is a neurotransmitter, is actually hindered. The neurotransmitter acetylcholine, it does what it is found in the nervous system and is there for transmission of impulses from the brain of the brain of the central nervous system to the body via the nerve cells. Then now we'll look at various conotoxins, whereby we find the W conotoxin, which targets the voltage-gated calcium channels, and K conotoxins, which target the potassium channels. We remember that what they do is that they just hinder the mechanism of closing and opening. This is whereby it is shown how the conotoxins affect the closing of the channels on the presynapses. Then we'll see that the effects of the corn snail conotoxins, they do what they cause severe pain, numbness, and tingling. And how do they do that? They do that by the blockage of channels, which results in what? The muscle paralysis, blurred or bivision. If you say bivision or double vision, which is double vision, and then the respiratory paralysis, meaning that you might find the hardness when it comes to breathing, and it indeed does lead to what? To death. Ladies and gentlemen, I thank you.